Welcome back. This video lesson is on DNA packaging and the learning goals to be achieved from this session are 1. Express the human genome size and number of base pairs in a typical haploid and diploid cell. 2. Explain how DNA is packaged inside the nucleus of eukaryotic cells. And 3. Differentiate between euchromatin and heterochromatin. Well, we know that DNA is a genetic material of living organism and is found in almost all the cells of an organism. The total amount of DNA or the total amount of genetic material in an organism is collectively referred to as genome. Suppose if we express genome size in E. coli in terms of number of base pairs, then the genome size in E. coli would be equal to 4 million base pairs. And if we express genome size in E. coli in terms of length, then the genome size in E. coli would be equal to 1.36 millimeter. However, the typical size of an E. coli cell is only 1 micrometer. Similarly, if we express the genome size in humans in terms of number of base pairs, in a typical human haploid cell, we have 3.3 billion base pairs. And a typical human diploid cell has 6.6 billion base pairs. And if we express human genome size in terms of length, then the length of human DNA is equal to 1.1 meters in a haploid cell, or it is equal to 2.2 meters in a diploid cell. Now in this connection, you can note a simple formula for the calculation of the length of DNA. Length of DNA can be calculated by multiplying total number of base pairs with distance between two consecutive base pairs. So now putting the values here, total number of a diploid human base pairs is equal to 6.6 billion base pairs, while we know that the distance between two consecutive base pairs is equal to 0.34 nanometer. And 1 nanometer is equal to 1 billionth of a meter. So now multiplying these, we get the product, which is the length of human DNA is 2.2 meters. While the typical size of a eukaryotic nucleus is only 10 micrometer. So how does such a long DNA molecule fit inside the tiny prokaryotic cell or a tiny eukaryotic nucleus? So the straight answer would be through DNA packaging. DNA packaging is an indispensable process of tightly packing up and organizing an organism's DNA molecule so as to fit inside the nucleus of a cell. Now, without going into the details, I want you to remember that in prokaryotes, such as in bacteria, DNA is packaged into a specific region in the cytoplasm called the nucleoid through supercoiling. This is because in prokaryotes, they lack a membrane-bound nucleus. Even though prokaryotes are not having a membrane-bound nucleus, their DNA is packaged or supercoiled with proteins and concentrated into a specific region called the nucleoid. It's apparent that such a long eukaryotic DNA cannot be existing as free linear strands. Eukaryotes have a complex DNA packaging strategy that exhibit different levels of DNA organization within the nucleus. Now in this lesson, we will focus on the three main levels of DNA organization, which are at the primary or the basic level of DNA packaging, DNA are organized into a structure called as nucleosomes or beads on string chromatin fiber. At the next level of DNA packaging, DNA forms an organized structure known as 30 nanometer thick fiber, or it is also sometimes called as solenoid. And at the highest level of DNA packaging, DNA forms an organized thick condensed structure known as metaphase chromosomes. So through these different levels of DNA packaging, DNA undergoes different degrees of compaction and condensation, where at the first level, at the nucleosome level, DNA is shortened by 7 folds. And at this 30 nanometer thick fiber level, DNA is shortened by 50 folds, whereas at this very highly condensed thick chromosome, metaphase chromosome level, DNA is condensed by over 10,000 folds. So such is the complex DNA packaging strategy that is employed by eukaryotic cells so that they can be easily fitting in the long DNA molecule inside the microscopic nucleus. At the most basic level of DNA packaging, known as nucleosomes or chromatin fiber level, negatively charged DNA molecule is wrapped around positively charged basic proteins in the nucleus called histone proteins, which results in the formation of nucleosomes. So nucleosomes are complexes formed by DNA and histone proteins. And each nucleosome is composed of a core of histone octamer, which is made up of 8 subunits, which includes 2 molecules each of H2A, H2B, H3 and H4 histone subunits, which is wrapped by approximately 2 rounds of DNA nucleotides, forming the nucleosome complex. And such a nucleosome complex is further sealed together or tightly stabilized together by a molecule of histone called as H1 linker histone. And in this way, the long DNA molecule is wrapped around hundreds of thousands of histone octamers, forming a long chain of or a repeating chain of nucleosomes. And such a long chain of nucleosomes or repeating units of nucleosomes are referred to as chromatin fiber. And a chromatin fiber has an approximate thickness or a diameter of 10 nanometer. Now, when a chromatin fiber is observed under electron microscopy, these chromatin fiber appear as beads on string structure, where beads represent nucleosome. So each bead in the beads on string chromatin fiber represents a nucleosome, and the string represents linker DNA or the DNA which is joining between the nucleosomes. So in this way, in the first level of or at the basic lowest level of DNA packaging, DNA is condensed or compacted up to 7 folds by forming repeating units of nucleosomes. At the next level of DNA packaging, known as 30 nanometer thick fiber or solenoid level, the long thin beads on string chromatin fiber is further coiled and supercoiled into a shorter thick fiber with a 30 nanometer diameter, and hence such a fiber is referred to as 30 nanometer thick fiber, or it's also sometimes called as solenoid. So such rounds of repeated coiling and supercoiling has resulted in further shortening of the DNA by 50 folds. At this point, I want you to remember that DNA will exist as chromatin fibers in all non-dividing cells or when the cells are in interphase stage. And these chromatin fibers are going to be distributed or scattered throughout the nucleus of a cell. However, depending on the level of compaction of chromatin fibers during interphase, they can be of 2 types, euchromatin and heterochromatin. Euchromatin are loosely packed region of chromatin fibers, and heterochromatin are densely packed region of chromatin fibers. In other words, euchromatin are the less compacted chromatin fibers, and heterochromatin are the more compacted chromatin fibers. I'll now explain some of the differences between euchromatin and heterochromatin. As I said earlier, basically they differ in terms of their compaction. Euchromatin is a loosely packed fiber, while heterochromatin is a densely packed fiber. And euchromatin has a 10 nanometer thickness, whereas heterochromatin has a 30 nanometer thickness. In other words, euchromatin is the beads on string chromatin fibers, while heterochromatin is the 30 nanometer thick fiber or the solenoids. And because euchromatin have a loose packing, so when these chromatin fibers are stained, they are going to be staining lighter, whereas these densely packed heterochromatic regions are going to be staining darker. And in terms of their location inside the nucleus, euchromatic regions are more towards the center of the nucleus, whereas heterochromatin fibers are lying towards the periphery of the nucleus. And in terms of their gene composition, euchromatin are the regions where we have active genes or the functional genes which can be expressed into proteins. Whereas heterochromatic regions have highly inactive genes, genes which are not expressed or occasionally expressed, or these are the regions where we have high repetitive DNA sequences. And because of the loose packing of the euchromatic fibers, the genes are accessible for proteins and enzymes, whereas because of the dense packing of the heterochromatic region, these genes are inaccessible for enzymes or for other proteins. And finally, we can differentiate euchromatin from the heterochromatin fibers in terms of their ability to transcribe into mRNA. So, euchromatin is the region which can be transcribed, or these region of the DNA can be copied into an mRNA. And hence these regions are known as transcriptionally active regions, where the DNA is copied into an mRNA and then the mRNA can be synthesizing a protein. Heterochromatin regions are the regions where the DNA will not be transcribed into mRNA because of the dense packing of that particular chromatin fiber. So DNA cannot be copied into an mRNA, and because the DNA is not transcribed into an mRNA, such region is referred to as transcriptionally inactive region. So most of the time heterochromatic regions are not transcribed, and because they are not transcribed into mRNA, so we cannot be expecting much of the protein synthesis from the heterochromatic region. Finally, at the highest level of DNA packaging, by using additional set of proteins which are collectively referred to as non-histone chromosomal proteins or NHC proteins, DNA molecule or the chromatin fiber is further folded into multiple loops and coils, and this results in the formation of the most compacted DNA organization called the metaphase chromosomes. And these metaphase chromosomes have a diameter or a thickness of 1400 nanometer. So such thick metaphase chromosomes have a discrete X-shaped structure which we can even see under a common light microscope. And at this highest level of DNA organization, DNA is compacted or shortened by 10,000 folds. That said, chromosomes are not always present in a cell. Chromosomes are formed in a cell around the time of cell division. And once the cell completes its cell division, its chromosomes are going to be decondensing back into the thin chromatin fibers so as to enable the cell to retain its ability to transcribe and replicate its DNA. By now, I hope you can imagine how much packing has happened in our long DNA molecule to fit inside a 10 micrometer nucleus. With this, I end my lesson on DNA packaging, and I'll come up with my next video lesson on DNA replication.